Hello, science class. I just wanted to say thank you for your patience with our class. And I also want to say just keep calm because you're an awesome class. We're going to get through this. All right, so today's class is going to be a brief recording. It's only going to be about 15 minutes. So let's get started. First, this was what I was trying to show you at the beginning of class. This is the way that our class will typically run. So over here, once you log in, will be a do now. The survey will stay the same, so if you want to bookmark it, that way you can access it easier, please do so. It's kind of to help guide my instructions. So the more thoughtful you are in your responses, the better I can help your facilitate your learning. It'll also have the agenda over here, what we hope to accomplish within the class. So you can see the do now. We're going to create a shared folder. We're actually probably going to skip reviewing section 1 and 2 because it looks like some students still need some time with it. We're going to cover the hypothesis and variables presentation, scientific methods questions practice you're going to do at home. I'm going to show you how to get started, as well as the controlled experiment. Your homework for tomorrow, or sorry, for next class, you're going to read chapter 1, section 3, here are the page numbers, and then complete that section review. I'd also like you to do the controlled experiment, and again, don't forget to complete your science method question practice. I'll update this. We're not going to do the Penny Lab just because I've spoken with your parents. We're going to wait till Tuesday after Labor Day for the Penny Lab because I want to make sure that we have a platform that's functioning before we try and do our first lab. All right, so another thing I wanted to remind you of and show you is our class homepage. All right, please, please, please make sure you highlight this page. I'm going to send it to you, or bookmark it rather. I'm going to send it in the email that's sending out in this with this recording. But you can click here. I don't know if we're going to use WizIQ. Likely we will be using Zoom, at which point, once I make that switch, I will also put that link right here until we know whichever link we're going to use. I will put it right here on the front of the home page. All right, remember announcements are over here. I'm not sure why that looks funny. I'll fix it. Your homework can always be found here. I'm going to update this. And then the materials for class, I'm going to update this as well. This was for today, but again, I want to get the recording out to you as soon as possible. Science assignments. So someone informed me that your calendar, you couldn't see items on your calendar. This is what your calendar should look like now. I think we fixed that problem. All right. Now then, you can also access all of the presentations. If you go under first semester, we're in chapter 1. And if you scroll down, everything you need to access and everything that I'm going to show you in class is under our chapter 1 or under each chapter. So for instance, the binder check with hyperlinks to all the assignments that we're going to that I'm going to be looking for and grading at the end of Unit 1 or at the end of Chapter 1 is right here. You can click on this hyperlink to access and open that document. Someone informed me that that you couldn't access the document, so I double checked and you are able to now. If you scroll down, here are all the presentations that we're going through. You can access those. We're actually going to be on this one today. And if you keep going, here's the activity zone. All right, you should have already completed graphing with POSI. We're going to do this scientific method questions practice today. You're going to do this controlled experiment over here and finish these two at home. And then for our next class, what we're going to do is we're going to do the science increments, so the System International, and we're going to complete that. We'll do the Penny Lab on Tuesday. I'll remind you as you go through. And then remember, there's helpful videos here. All right, so let's get started with our presentation today. I'm going to skip those review slides, but they are there for your perusal. So if we're looking at these statements, the students were having a discussion about the upcoming science fair, and what we want to know is, are the students posing a testable question? So for instance, Julian, is it more exciting to drive a motorcycle blindfolded or go skydiving? Hopefully Julian is not going to test natural selection and ride a motorcycle blindfolded, but we ask the question, is it observable? Yes, it's observable. Is it testable? It might be, but how are you going to measure exciting, the level of excitement? So that one's kind of an interesting one. Melissa, is Einstein the greatest scientist ever? Well, we ask ourselves the question again. Is it observable? No. Is it testable? No, not really. What about Jason? How do different types of activity affect human heart rate? That one, is it observable? Absolutely. Is it testable? Absolutely. One of your classmates, Daniel, made the suggestion we could test something like if a human sat and played a horror game show. Uh, then you could test whether or not the heart rate changed when they're playing this horror game. And you can see how that would vary. So it's definitely observable and it's certainly testable. Antonella, why is the sky blue? Well, it's observable and to some degree it is testable. So you should always be asking yourself, is my hypothesis observable and is it testable? So with that, we're going to get to questions, hypotheses, and variables. So again, here's that focusing on the testable questions. So a testable question is one that we can answer by designing and conducting the experiment. They have to be observable and they have to be measurable. Those are the two criteria. Well, a hypothesis is a testable explanation. I know many of you have probably been told that it's an educated guess. You were lied to. It's not an educated guess. It's a statement that you make based on research, based on observations that you're able to test. All right, so elements of a hypothesis, it has to define the proposed relationship between the variables. So we're looking at a cause and an effect. Hypotheses are always worded in an if-then statement. If I play a horror game, then my heart rate will increase. It's something that's observable and it's something that's testable. It has to be worded again so that you can make sure that it's testable. And it needs to make a clear prediction. I know I'm making a prediction about what's going to happen. I may not know what's going to happen, but I can make that prediction. And it needs to be specific. Within any experiment, we have variables and controls. A variable is anything that's going to change in an experiment. It's the thing that we're manipulating. So maybe in the game example, we're manipulating, we're saying if you play the game, then your heart rate will increase. So our variable would be playing the game or not playing the game. The control is what you compare your results to the end of the experiment. So in that example that your classmate proposed, we would need to design an experiment where we all played the horror game. So we would all not play the horror game, and then we would all play the horror game. So for instance, if I am not playing the horror game, so we're all going to not play the horror game and we're going to record our heart rate for 5 minutes. That's our control because nothing's happening. So we have a baseline of what our heart rate should be. Then we're all going to play the game for 5 minutes and we're going to see how our heart rate increases. That's our variable. So with variables, there are two different types. We have the independent variable, also known as our manipulated variable, and we also have the dependent variable, known as our responding variable. The independent variable is the variable that we're manipulating. We graph it on the x-axis and it's our cause. So let's say we're doing an experiment with effervescent tablets, and maybe our hypothesis goes something like this. If I increase the water temperature that the effervescent tablet is dropped into, then the effervescent tablet will dissolve faster. All right, so I'm manipulating the temperature. The dependent variable then would be how fast that effervescent tablet is dissolving. So the dependent variables are responding variable, it's the effect, and we're graphing that on the y-axis. It's what we're actually measuring. Okay, so in this case, it's time. To help you graphically, so data tables or organizing your information into data tables always helps. So you can see here we've got the independent variable. It's also known as the manipulated variable. It's our cause and our cause-effect relationship, and we always plot it on the x-axis. The dependent variable, on the other hand, is our responding variable. It's what gives us our effect, and it's always graphed or plotted on the y-axis. All right, now there's constants or controlled variables. And these are things that, items that are kept the same through each trial in the repeated experiments. So for example, in our effervescent tablet dissolving by temperature, well, things that I would want to hold constant is for each experiment or for each trial, I would want, let's say we're going to do 3. Let's say I have one at room temperature, 1 beaker at room temperature, 1 beaker at, let's say, 40 degrees Celsius. And let's say I have a third beaker that's really hot, and that one's going to be maybe at 60 degrees Celsius. So the amount of water that I put in each beaker, I would want to hold constant. The brand of the effervescent tablet, I would want to hold constant. The size of the effervescent tablet that I put in, whether or not I stir it, those are all things that I want to keep the same for each of them because those are variables that could affect the outcome as well. So you want to try and control as many variables within the experiment as possible. We only want to see what our independent variable or our manipulated variable is doing. All right, so a control group, on the other hand, is something that it's the standard for which we're measuring our experimental groups against. So I'll go back to the class example where we're playing a horror game. Then when we sit for 5 minutes and take our heart rate for 5 minutes, that's our control group in that experiment. Us playing the horror game is our experimental group. I need a baseline measure. With our effervescent tablet experiment, the room temperature water is our control group. It's what we're measuring that 40 and 60 or middle and higher temperatures against so that I know how it's supposed to respond or if there is a response rather. All right, so here's another example. Let's say you're planting tomato plants and you think that instead of water, you think that some other type of liquid is going to make the tomato plant grow better. So things that we'd want to hold constant or our controlled variables. I'd want to make sure that the size of the pot for each of my trials are the same. I would want to make sure that the amount of soil and fertilizer, if I'm using fertilizer, would be the same in each one. I'd want to make sure that I'm using the same type and brand of tomato seed for each one. Now then, my independent variable is the actual liquid that we're using. So whether or not we're using soda, juice, or water. And then the dependent variable, what we're actually measuring as a result of this, is the height of the plant. Okay, and in this experiment, the one with the water is our control group, and these two, the soda and the juice, are our experimental groups. All right, so you can see this. We can break down a sentence. So if fertilizer helps grow larger plants, there's my relationship between the variables. And a plant is given fertilizer, here's my manipulated variable. So I'm either going to give the plant fertilizer, that would be my manipulated variable, or I'm not, which would be my control group. Then it will be larger. And here's my responding variable. So that's what I'm going to plot on the y-axis, and I can measure the height of the plant. All right, so let's practice with the scientific method questions worksheet. That's what you're going to do independently. So let me just show you what that looks like. Okay, so that's this worksheet here. Again, you can find that on our website. Sorry. Here's our website, and it's right here. It's in the activity zone, right there under the Graphing with Posey, which you should have already completed the Graphing with Posey. So on the scientific method questions practice, read the background, and then here are your directions. We'll go over this on the next class. I'd also like you to go to the controlled experiment. That's right here again under the activity zone. Once you click on that link, you're going to get this virtual experiment. So read the directions, they're pretty straightforward, and see if you can run the controlled experiment so we can practice, and I will see you on the next class. I'm going to send out all the information. If we use Zoom, I will send you the new hyperlink. I will also post the hyperlink on our website on the home page where the WizIQ is. Thank you so much. I appreciate your patience, and I will see you on Thursday.